Imagine watching a generic, uniform ball of human cells, like nothing special about them at first glance.
Right, just a complete blank slate.
Exactly. But then, as if following some invisible ancient set of instructions, they just decide to organize themselves.
All on their own.
All on their own. They start building a structured body plan. They lay down the very first blueprint for organs. They start forming the earliest blood cells. And they do all of this in a lab dish without ever using a real human embryo.
I mean, it sounds like pure science fiction, honestly.
Mm-hmm.
But it is actually one of the most profound biological realities we are currently exploring.
It really is.
We're talking about recreating the absolute earliest, most secretive moments of human development right before our eyes.
And that is exactly the mission of today's deep dive. We are looking at a fascinating 2026 technical report. It was published in Nature Cell Biology by Jianping Fu and a team of researchers.
Yeah, a really groundbreaking paper.
They've created something called the PTED embryoid. It is this brand new lab-created model that perfectly mimics a highly critical yet incredibly mysterious window of human development called Peri-Gastrulation.
Which is a bit of a mouthful, I know.
It is. So, okay, let's unpack this.
To really understand the magnitude of this paper, I mean, we have to set the stage with the actual stakes involved.
Right. Why is this so hard to do?
Well, historically, studying this specific early phase of development in actual humans has been practically impossible. We call it the black box of human development.
Because it's completely hidden.
Exactly. By the time the peri-gastrulation phase occurs, a natural embryo has already implanted into the uterine wall, so it is completely hidden from view.
Right, so we literally cannot watch it happen in vivo.
No, we can't. And ethically, studying natural human embryos in a lab at this stage is incredibly fraught.
Oh, absolutely. There are strict global limitations on that.
Exactly. So scientists have essentially been operating in the dark, just trying to guess how the foundation of the human body is actually built.
Which is where this new model comes in.
Right. This new PTED model completely bypasses those technical hurdles. It gives us a clear, unobstructed window into our earliest beginnings.
Which is just wild to think about. We finally have a flashlight to look inside that black box.
A very bright flashlight, yeah.
But to really get why this lab model is such a massive breakthrough, we need to understand the biological event it is trying to mimic. So let's talk about peri-gastrulation.
Sure.
What exactly is happening in this blueprint phase?
So peri-gastrulation is arguably the most dramatic physical transformation in a mammal's life.
It's the big one.
It really is. You start with this homogeneous, uniform population of cells called pluripotent epiblast cells.
And pluripotent just means they have the raw potential to become almost any cell type in the body, right?
Exactly. They are basically blank slates. But during peri-gastrulation, these generic cells suddenly undergo this massive event of self-organization.
They stop being generic?
Right. They establish the fundamental body plan. Specifically, they form something called the trilaminar embryonic disc.
The trilaminar disc, meaning 3 layers.
Yes. The embryonic ectoderm, the mesoderm, and the endoderm.
Got it.
And these 3 layers are arranged along a dorsal-ventral axis, which is basically a top-to-bottom orientation.
Okay, so what do those layers actually become?
Well, the ectoderm will eventually become things like your skin and your nervous system.
Ah, okay.
The mesoderm becomes your muscles, bones, and blood. And the endoderm becomes your gut and internal organs.
Okay, for everyone listening, let's make this really visual. Imagine a perfectly constructed sandwich suspended right in the middle of a balloon.
I like this analogy.
Right. So the sandwich in the middle is that trilaminar embryonic disc we just mentioned, the 3 layers of meat, cheese, and bread that make up the actual body.
Exactly.
Now, the top half of the balloon, covering the dorsal or backside of the sandwich, is the amnion. That's a protective fluid sac.
Yeah.
And the bottom half of the balloon, on the ventral or belly side, is the definitive yolk sac.
That is a highly accurate structural analogy. But what makes this so incredibly complex is the communication required to build it.
Right, because nobody is telling them what to do.
Exactly. How does a uniform spherical ball of cells suddenly know to flatten out and separate into a sandwich suspended between 2 balloons?
It's crazy to think about.
How does a cell know that it needs to become part of the yolk sac at the bottom rather than part of the neural ectoderm up at the top?
So how does it happen in nature?
In nature, inside the womb, there's a highly complex signaling cascade. It's a symphony of chemical signals telling cells exactly where to migrate, when to multiply, and what to become.
So to put this in perspective, in just over a week of natural development, we go from a uniform puddle of cells to a highly structured 3D anatomical model.
It's an incredibly tight timeline.
Let's quickly recap what we've established so far. Time for our first quick summary. Number 1, Peri-Gastrulation is the crucial body blueprint phase. Number 2, it forms that 3-layer disc we talked about. And number 3, this disc is sandwiched between the amnion above and the yolk sac below.
Perfect summary.
Think about your own body right now, you listening. Does it blow your mind that every organ you have started as a single flat disc of cells sandwiched between 2 tiny fluid sacs?
It really is awe-inspiring when you stop to think about it.
It really is. And that brings us to the next massive challenge. If that is what peri-gastrulation looks like in nature, how on earth did these researchers recreate it in a petri dish?
That is the million-dollar question.
Let's get into the recipe. How did they build the PTED model? And just to clarify, PTED stands for Peri-Gastrulation Trilaminar Embryonic Disc.
Correct.
What exactly were the ingredients here?
So the researchers started with a very specific foundation. They used primed human pluripotent stem cells.
Okay.
Specifically, a well-known, widely used cell line called the H9 line. These are cells that are developmentally primed to mimic the early epiblast we just talked about.
Makes sense.
But here is the critical part, and the paper emphasizes this heavily. The model is completely transgene-free.
Wait, I want to push back on that for a second because that sounds like a bit of technical jargon.
Sure, let's break it down.
Previous embryoid models often used genetic tweaking, right? Like they used transgenes to essentially hack the cell's DNA and force them to change into specific tissues.
Yes, exactly.
But this paper proudly plants its flag on being transgene-free. Why does that distinction matter so much?
It matters immensely because of physiological accuracy.
Okay, how so?
Well, when you use transgenes to force gene expression, you are essentially hijacking the cell's internal programming. You are chemically dragging it down a path.
Right, you're overriding the system.
Exactly. And the problem is that forced expression can activate unintended genetic programs.
Oh, I see.
It can trigger unnatural biological pathways that would never, ever actually happen in a real human embryo.
So it ruins the whole point of making an accurate model.
Right. A transgene-free model, on the other hand, relies entirely on pure self-organization.
So we aren't writing new code for them. We are just giving them the right environment to execute their own natural code.
Precisely. It means the cells are doing the heavy lifting themselves, using their own innate biological memory.
Which is way more natural.
Exactly. Which makes it a vastly more accurate representation of natural human biology.
Okay, but if we aren't forcing them genetically, how did the researchers coax these cells into building the sandwich inside the balloon? What was the actual mechanism here?
It came down to a combination of exact physical geometry and a very brief chemical trigger.
Geometry, like shapes.
Yes. The researchers took exactly 5,000 of these stem cells per square millimeter and placed them onto tiny circular adhesive islands on a glass cover slip.
Tiny islands.
Yeah, these islands were exactly 800 micrometers in diameter.
Wow.
To picture that, it's less than a millimeter across. And they were coated with a supportive substance called Geltrex.
Why 800 micrometers specifically? That seems incredibly precise. Is there something magic about that size?
What's fascinating here is that the size and shape dictate physical tension.
Tension, like they are stretched.
Kind of, yeah. Geometric confinement physically forces the cells to interact with each other in a specific spatial pattern.
Oh, I see where this is going.
Think about it. A cell sitting right on the outer edge of that tiny circle is experiencing different mechanical stress and tension than a cell sitting perfectly in the center.
Because the central cell is surrounded by neighbors on all sides.
Exactly.
Ah, so it's not just about keeping them contained. The physical crowding actually changes how they behave.
Precisely. It's a process called mechanotransduction. The physical forces literally change how the cells interpret chemical signals.
That is incredible.
Once the cells were confined in this exact pattern, the researchers added a single chemical signal, exogenous BMP4 or bone morphogenetic protein 4.
Okay.
And they only added it for exactly 2 days.
2 days. So think of the geometric confinement like putting 5,000 strangers into a perfectly circular, walled-off stadium.
I love that analogy.
And the 2-day BMP4 pulse is like a single loud whistle blow.
Right.
The shape of the stadium forces the crowd to organize based purely on who they are standing next to and how tightly they are packed together when that whistle blows.
That is a phenomenal way to visualize it. The cells on the edge, they hear the whistle differently than the cells in the center because of that physical crush.
It's all about context.
Right. And after day 2, the researchers stopped blowing the whistle. They completely withdrew the BMB4.
But the damage was done, or rather, the instruction was given.
Exactly. The cells had already received the message. They began to divide, differentiate, and spontaneously form the complex 3D structure entirely on their own.
It's essentially like tapping the first domino. The chemical doesn't build the structure, it just tells the cells to wake up and start building it themselves.
That's a perfect way to describe it.
So if we take a step back and look at what we've covered so far, let's do our next quick summary. Number 1. PTED relies on primed stem cells. Number 2, no genetic modification is used. It is pure chemical and physical prompting.
Transgene-free.
And number 3, a 2-day pulse of BMP4 on tiny adhesive islands kicks off the magical self-organization.
That's the recipe.
I want to ask you listening right now, if you were given exactly the right chemical signal and the perfect physical space, what kind of hidden potential do you think you'd self-organize into?
It's a great philosophical question.
Isn't it?
And biologically, the answer in this experiment is just staggering. Let's look at the resulting architecture. We've applied the physical tension. We've tapped the first domino.
What actually emerges by day 8 of this experiment?
The day 8 results are honestly a marvel. Over those 8 days, these flat, 2-dimensional puddles of cells actually detach from their glass cover slips.
Wait, they pop off the glass?
They do. They become completely free-floating, polarized 3D structures. They literally build their own 3-dimensional space.
That is mind-blowing. But I have to ask, how do we know they actually built the right things?
What do you mean?
Well, you can't just look at a microscopic floating bubble of cells and say, yep, that's a yolk sac. They all just look like mush, right?
You're absolutely right. Visuals aren't enough at that scale.
So how did they prove it?
That's where we look at the molecular ID badges. The researchers needed to prove that the cavities and layers they were seeing weren't just, random disorganized tumors.
Right.
They had to prove they were actually the precise, highly specific tissues found in human peri-gastrulation. So they used advanced immunostaining.
To look for what?
To look for permanent molecular barcodes,
Yeah.
specific proteins that only certain cells produce.
Let's walk through that anatomy using those molecular barcodes, going back to our sandwich and balloon analogy.
Okay, sounds good. Starting with the top balloon, the dorsal cavity.
The amnion.
Right. The researchers probed the cells lining this top cavity and found the exact molecular signatures of non-neural ectoderm and amnion cells.
What were the actual markers?
They looked for markers like TFA-P2A and ISL1, and the genetic barcode perfectly matched. So that confirms the top cavity is an authentic amniotic-like sac.
Check. Top balloon is the amnion. What about the sandwich in the middle?
The central dense region, the trilaminar disc, showed the coarse organization of 3 distinct layers.
The meat, cheese, and bread.
Exactly. They found specific protein markers, like SOX2, confirming the embryonic ectoderm on top.
Okay.
They found classic markers for gastrulating mesoderm, like BRA, right in the middle.
Yeah.
And they found the definitive barcodes for embryonic endoderm, like FOXA2, on the bottom.
Check. The sandwich has its 3 distinct layers, and the bottom balloon.
The ventral cavity at the bottom was enclosed by a single layer of cells.
And what did its barcode look like?
It was expressing a very specific combination of molecular tags, FOXA2, SOX17, HNF4A, which proves it is extraembryonic endoderm.
In plain English?
In plain English, that means it is a definitive yolk, sac-like structure.
Here is where it gets really interesting. All the structures are there, the barcodes perfectly match. But the source paper highlights a massive biological plot twist regarding where these structures actually came from. It has to do with something called lineage tracing.
Oh, yeah. This is perhaps the most significant scientific paradigm shift in the entire paper.
Seriously, why?
Well, for context, in developmental biology, there has been a long-standing assumption about when extraembryonic tissues, like the yolk sac, diverge from the embryonic tissues that actually make the body.
Why was there an assumption at all? What was the old dogma?
The traditional thought was that these lineages separated very, very early on, like almost sealing off their fates from one another.
So they branch off and never interact again.
Exactly. And the logic made sense. Building the support structures, like the placenta and yolk sac, requires incredibly fast, aggressive cellular growth.
And fast growth means mistakes, right?
Precisely. Rapid division is prone to genetic errors. So the theory was that the embryo locked down the cells meant for the actual body early on.
To protect them from mutations.
Right, while delegating the heavy lifting to a completely separate branch of support cells.
Got it. Like totally different branches on a family tree that split generations ago.
Exactly. But to be sure, the researchers used a sophisticated lineage tracing tool. Think of it as a permanent molecular tracking device.
How does that work?
They essentially put a dab of glowing genetic paint on a single gastrulating ancestral cell in the central epiblast.
Just one cell.
Just one. And every time that cell divided, it passed that glowing paint to its daughters.
Oh, that's clever.
It allowed the researchers to literally watch exactly where every single daughter cell ended up.
And what did the glowing paint reveal?
It completely overturned the old dogma. The lineage tracing proved that both the embryonic tissues, the mesoderm and endoderm of the actual body, and the extraembryonic tissues, like the yolk sac, arose from the exact same gastrulating cells.
Wait, they came from the same source?
At the
exact same time.
At the exact same time.
The central epiblast cells gave birth to both the internal body layers and the external yolk sac.
Precisely. And if we connect this to the bigger picture, it solves a major biological puzzle. It proves that the human peri-gastrulation epiblast is incredibly flexible. It isn't locked into a rigid path early on. It actually retains the potential to create extra embryonic support structures much later than we ever thought possible.
It has the plasticity to build both the embryo and its support structures simultaneously.
Exactly.
That completely changes how we view stem cell plasticity. So, let's take a breath and do our Day 8 check-in. Our third quick summary.
Let's do it.
Number 1, the model successfully forms the amnion, yolk sac, and trilaminar disc.
Check.
Number 2, specific cellular markers prove the identity of each layer.
Check.
And number 3, the central epiblast cells surprisingly give birth to both the body layers and the external yolk sac.
Perfect.
Here's a question for you listening. How often do we assume a rigid, predetermined path for something, only to discover it has the flexibility to become several entirely different things at once?
It's a great lesson in adaptability, really, both biological and conceptual.
For sure.
Now, we have established that the PTED model is structurally accurate. It has the right architecture.
Right.
But there is a massive difference between building a structure that merely looks like a yolk sac and building one that actually acts like one.
Oh, absolutely. It's like building a perfect replica of a Ferrari engine out of clay.
Yes.
It looks great on a pedestal, and the dimensions are exact, but if you turn the key, nothing happens.
Exactly.
So does this lab-grown yolk sac actually turn on? Because this brings us to the most dynamic, frankly, jaw-dropping discovery in the paper, the blood factory.
Right. To understand this, we have to look at the true function of the definitive yolk sac in a real human embryo.
What does it normally do?
Well, before the fetal liver or the bone marrow develop and take over, the yolk sac is the very first site of blood formation in a developing mammal.
It's the original blood factory.
Exactly. This process is called primitive haematopoiesis.
And the researchers observed something incredible happening with the PTED yolk sac under the microscope.
They did.
It wasn't just a static bubble of cells sitting there like a clay engine. The source notes that this ventral yolk sac structure was exhibiting dynamic physical behaviors.
It was moving.
Right. It was periodically expanding and collapsing. It was physically pulsing, mirroring the actual mechanical behaviors of these tissues in real human development.
And that physical pulsing is a sign of immense underlying biological activity.
It's not just twitching randomly.
No, not at all. When they analyzed the tissue surrounding this pulsing yolk sac, they found the ancestral stem cells of the blood system.
What are those called?
Hematopoietic endothelial progenitors, or HEPs. They specifically found cells marked by CD34 and VECAD.
Okay, so finding these specific progenitor cells was a massive clue that active hematopoiesis, the manufacturing of blood, was gearing up.
Exactly.
But they didn't just stop at finding the progenitors, right? Finding the raw materials is one thing. They ran a fascinating physical test to see if the structure would actually finish the job and manufacture real blood.
They did. To truly prove function, they performed a bisection experiment.
Which sounds intense.
It's very delicate work. On day 8, they carefully sliced the microscopic PTED embryoid in half.
Just cut it right down the middle.
They separated the top half from the bottom half. They took that ventral half of the piece containing the pulsing yolk sac structure, and they cultured it entirely on its own in a specialized medium until day 14.
They literally cut it in half to isolate the engine. And what happened by day 14?
It worked. By day 14, that separated, isolated yolk sac tissue had generated real early blood progenitors.
Wow.
They found distinct colonies of cells with the definitive molecular signatures of early blood cells. They saw cells expressing markers like PU.1 and RUNX1.
So wait, let me get this straight. In a glass dish, using only a tiny drop of chemicals, some stem cells, and a little bit of physical tension, they managed to kickstart a biological engine that actively manufactured primitive human blood cells.
That is exactly what they did. This isn't just a structural model. Seeing active haematopoiesis happen in a lab dish is the ultimate proof of concept.
It really is.
It confirms the functional authenticity of the PTED model. It isn't a clay engine. It is a working, functioning biological machine that faithfully replicates the earliest blood-forming processes of a human being.
I am genuinely floored by that. It is astonishing.
It's a huge milestone.
So as we round out this deep dive, let's look at the full scope of what this team accomplished. Our final recap. Number 1, the PTED yolk sac physically expands and collapses.
Check.
Number 2, it successfully produces early blood cell progenitors.
Check.
And number 3, this proves the model isn't just structurally accurate, but functionally active.
Perfectly said.
So think about this. What stands out to you? Is it the perfect structural precision of the 3 layers forming entirely on their own, or the fact that real primitive blood cells are being manufactured from scratch in a laboratory dish?
It is a massive amount of information to synthesize, which brings us to the ultimate question regarding the source material.
Right.
We have this incredible model. We have proven it works structurally with the exact molecular barcodes, and we've proven it works functionally by producing early blood. But why is this so vitally important for the future of science and medicine?
Yes. So what does this all mean? What is the big takeaway for humanity here?
The ultimate value of the PTED model is that it gives us a highly tractable, incredibly detailed window into early human development.
Without the ethical baggage.
Exactly. It does so while remaining an ethically safe sandbox. This is a crucial caveat that is explicitly detailed in the paper. The PTED embryoid is not a viable embryo.
Right, because of what it's intentionally missing.
Exactly. Because of how it is formed from prime stem cells, the PTED model develops completely without 2 very specific lineages, the trophectoderm and the hypoblast.
What do those do normally?
Well, in a real human embryo, those specific tissues are absolutely required to implant into a uterus and to form a placenta. Because the PTED model completely lacks them, it bypasses the blastocyst stage entirely. It literally cannot implant, and it biologically cannot grow into a human being.
So it's an ethical sandbox where we can study the roots of congenital diseases or figure out why some pregnancies fail in those critical first few weeks.
Right.
Or even understand how to better generate blood and tissues for regenerative medicine, all without crossing the ethical lines associated with using real human embryos.
Precisely. It allows us to decode the earliest, most secretive chapters of the Human Instruction Manual safely and accurately, lifting the veil on that developmental black box.
And what a remarkably elegant instruction manual it is. We've seen today that human stem cells possess an innate, almost architectural memory.
They really do.
They don't need us to hack their DNA. They are able to build their own scaffolding, carve out their own cavities, and fire up their own pulsing blood factories with just a tiny bit of physical tension and a gentle chemical nudge.
It's beautiful, really.
So I leave you with this final thought. If generic cells know how to build the foundation of a human body all on their own, without a blueprint, what does that tell us about the deep encoded intelligence hidden inside the simplest building blocks of life?
That is something to think about.
Thank you for joining us on this deep dive into our earliest beginnings. Keep questioning and keep exploring.